but I'm thrilled to be here, and and thanks to Guy and the organizers for giving me the opportunity. So I'm going to keep things pretty high level, but what I'd like to give people a sense of is what Ovid is, some of the conceptual things that led to its founding, and then a little taste of the biology underlying this upcoming trial in Angelman syndrome, and then following that, just where we're at in terms of the actual trial. So I really don't probably need to emphasize this to anyone here, but of course, neurodevelopmental, sure, is that better? It's so loud up here, but I guess it'll work. So neurodevelopmental disorders are debilitating, of course, right? We see them all around us, and whether the rare disorders we're working on are AS or 15q dupe, they're numerous and come at an enormous cost both to families and to the society in terms of financial burden. So Ovid was really founded to address this. There's surprisingly little in terms of commercial work in this space, and so we see that as a real opportunity and a way that we can impact and hopefully make a change for the better. And I think this is a concept that really emerged in some of the talks earlier today, that regardless of the genetic etiology of any given disorder that one may be studying, in terms of a very high-level presentation, there are a lot of shared deficits. Now, of course, when we dig a little deeper, either in terms of clinical presentation or mechanisms, they diverge, certainly. But the hope is, at Ovid, that we can capture some of these overlaps and use that as entry points for different disorders. Something that folks have discussed here is, how incredibly difficult it would be were every single genetically defined disorder to require a completely unique and independent treatment, right? So, as Ben mentioned yesterday, each compound that makes it into the clinic, hundreds of millions of dollars of investment, if not billions of dollars, or $1 billion or so dollars, and a decade or more of work. So there really is this real desire to ensure that we can recognize all of these distinct etiologies in the ways that they're different, and then really take and leverage where they do overlap. Sorry. So this is a slide from many years ago, but it really kind of speaks to that same idea with a little bit more meat on it. So it's a hypothetical example, but you can imagine that various disorders at different parts of the genome, whether you're measuring gene expression or some cognitive ability or an EEG-related phenotype, that they may well converge, and that looking at those areas of convergence and defining a signature for individual disorders will really enable that entry point into a shared point of intervention. So I think with those ideas in mind, that this idea that we want to really capitalize around this convergence, seek overlap, and use that to drive forward targeted treatments, that's what really brought Ovid into being. And alongside of that insight, there were a number of factors independent of the conceptual thing that really brought Ovid to be, which is genomics, really identifying these genetic subgroups and enabling the kind of mechanistic study that we really think is essential for drug development, social media that has really enabled communities to be formed and come together in ways that were never before possible, and of course, this incredible mechanistic insights that have really emerged relatively recently and are going to be the foundation for all of the work that we pursue. So we've heard a lot this afternoon about GABA, but really at quite a high level. So we've heard the word many times, but really not dug in to it. And it's a very, very complex system, right? So central to the work that we're pursuing is that there's actually two functionally distinct classes of GABA receptors. You have these synaptic GABA receptors, and they mediate phasic excitation, And we've seen a lot of these traces that look like this. What we haven't really discussed, and is our interest, are these extrasynaptic GABA receptors that mediate a very different kind of neural activity, this tonic inhibition, okay? So it's critical for what I'm gonna talk about to distinguish between these synaptic and extrasynaptic receptors that are actually comprised by different subunits encoded by separate genes that come together in various combinations and give rise to different functional properties. And I think something else I'll mention here is that in the context of discussion of GABA today, there's been a lot of mention of benzodiazepines. The specific receptor subtype that we're targeting, these extrasynaptic receptors, they don't actually carry the subunits that enable binding of GABA, so that's entirely distinct from the approach that we're pursuing. So I basically said in the last slide, you have these synaptic and these extrasynaptic receptors that mediate these very different kinds of currents. And at a very high level, one way that you can think about tonic inhibition, if you're not an electrophysiologist, which I'm definitely not, there's some papers that are out that talk about tonic inhibition as signal to noise, right? So all of us are easily able to identify red from blue in this panel on the right, and although the red dots are not altered in this panel on the left, the presence of this background information makes that task very difficult to complete. And so this tonic inhibition, there's some data to suggest that at a very broad level, what it's actually mediating is this ability to identify signal in the background of noise. So just to dig into it a touch more, in each of these traces, and we'll focus on the left because those are the wild type animals that are being looked at, there's been a GABA antagonist that's been added on here, right? And here. And tonic inhibition, to actually visualize it in this way, you have to add on one of these antagonists, and the extent of tonic inhibition is reflected by this distance. Okay? So it's very much unlike standard recording, where you really see what's happening in real time. It's more deductive to be able to visualize it, relatively speaking. So relevant to our discussion here is that in the UBE3A Angelman model, you see that this distance relative to the wild type animal is diminished, and so tonic inhibition is reduced. Okay? And interesting, and kind of stepping back to our idea that there may be these shared deficits across disorders with very distinct genetic etiologies, but perhaps shared molecular mechanisms, the same diminished tonic inhibition is evident in the Fragile X mouse. Okay, so that's quite exciting, and at least at the level of electrophysiology, suggests that perhaps there's some ability to create an intervention that may be useful in each of these indications, right? And what, of course, conceptually that's very nice, but in the absence of a compound that could actually be taken to the clinic, you're stuck. But what is very valuable here is that OV101, also known as THIP, also known as gaboxidol, very specifically binds these extrasynaptic receptors, these delta subunit containing receptors that mediate this tonic inhibition. And it's the only known agonist specific to this receptor population. And so data from animal studies shows that it can actually increase this tonic inhibition, and in the Angelman mouse, normalize behavioral features that are observed. And so conceptually on the right here, if tonic inhibition is compromised in an animal model or presumably in patients, the ability to increase that potentially through treatment with OV101 would improve or perhaps normalize function. And maybe I'll just say as well, if we contrast with a gene therapy approach, this is downstream of UBE3A, of course. So we're not at all trying to influence or expect re-expression of UBE3A, but we see this as a very complementary and necessary strategy in that there's data out there from Ipe Elksmer's lab that suggests that even complete reactivation may not be sufficient to enable normalization of function. So this downstream approach for Angelman and in other disorders, I think, may be quite valuable. So here's some of the behavioral data. What we're looking at here on the left are mice stepping along a walkway with painted feet. And it's a little hard to tell that there's actually any difference here in that the step length is normal, but if you actually look closely and the direction at which these paw prints fall, there is an increased abduction or outward positioning of the feet in the Angelman mouse. And that's actually normalized in the presence of THIP, which is OV101, which is this compound that we're taking to the clinic. And the same is true in the Fragile X model. Separate work shows that THIP is able to normalize this gait deficit, this motor deficit. And so it's always difficult, of course, to know how work that's observed in an animal model might translate to the clinic. But based on these and other data, we're optimistic that motor features, and in particular gait, may be something that's ameliorated. And that's quite exciting in that, a difficulty in all of these trials, and we've heard this several times, is that some of the higher order cognition, language aspects, may be quite difficult to capture in a quantitative way, and as well may require a longer period of treatment relative to motor deficits. And so the fact that we actually see in the animal models an amelioration of some of the motor phenotypes is quite promising. Other data that I'm not going to talk about, but likewise gives us hope, is that actually neuronal maturation is impacted positively by treatment with THIP, and it's actually also found to improve sleep architecture. It was actually, I'm going to mention in a moment, but it was initially developed for treatment of insomnia, and so particularly in AS and other neurodevelopmental disorders where sleep is such an issue, it may well have benefit there as well. So in terms of the trial, things are, I think we're set up nicely to move this forward. And another element that's really, I think, really drove Ovid forward is the idea that rather than starting from scratch in a discovery space, looking for compounds that may be able to normalize dysfunction in quite interesting pathways, we look downstream at compounds that we know are safe, we know are well tolerated, and begin there and really capitalize off the enormous investment that's already been spent. So OV101 or gaboxidol has already been evaluated in more than 3,000 individuals. It was essentially, so it was developed by Lundbeck and Merck, and a lot of investment spent there. So we know that it gets to the brain, and that the pharmacokinetics also look good, at least in adults. And so because we do not yet have data, pharmacokinetic data from pediatric patients or adolescents, this trial that we'll launch in the fall will be in adults. And the primary goal here will be to assess safety and carry out some exploratory work to evaluate those measures that might be most informative in a phase 3 trial. Okay, so sites are just getting underway, but we're incredibly fortunate to have folks in this room with so much expertise guiding us on the design of the trial and enrolling patients hopefully in the near future. And so I think I will stop there. One thing I will mention is that things it is a pretty exciting time for us. We're looking at a variety of other indications and opportunities there, and additional compounds that we think may be useful. So hopefully at the next meeting I'll be able to talk about some of that work that we'll be able to have moved forward. Thanks. Yeah. Sure. So Merck and Lundbeck initially developed this for insomnia, and the idea was that Ambien is a scheduled drug, that perhaps this could be brought forward unscheduled. And at the end of the day, despite good data showing efficacy in prolongation of sleep and actually with good preservation of architecture, there was no obvious benefit relative to Ambien. And so from a business context, you had such market saturation of that drug, there was no way that this was going to go forward and be a commercial success. And so that's the rationale for them not pursuing it.